mapping of the microRNA expression. And in this case, it's about the cardiac development. This is something that a lot of people are interested in listening. It's a new technology, it's very hot, but it doesn't work in the hands of everyone at the moment, but is part of the future technology. So we are really interested in hearing your experience and what you have accomplished so far. Stefanos, the virtual space is yours.
Thank you very much, Eric. I hope everyone can hear me. I see that the screen sharing is disabled, though. Can we enable that?
It should be working now.
Let me see.
Oh, wait. Yes.
Okay. So, okay. So, hello everyone, I'm Stefanos. I'm in the lab, the genetics lab in the Agricultural University of Athens, working with Mitsis. I'm a postdoctoral researcher, and right now I would like to talk to you a bit about single cell mapping of microRNA expression in cardiac development. Now, this is some work that has started back in Sweden, actually, and we've been continuing it here in Athens, in the Agricultural University of Athens. And I think it's a very exciting thing, and we are trying a quite novel approach to decipher the microRNA expression in a single cell model. Now, why in the cardiac development? The thing is that cardiovascular disease, which is quite a broad term, is actually one of the main cause of death worldwide. At least it was until just before COVID. So I don't know the current situation. I've heard that COVID had some effect in the admittance of people suffering from either some kind of extreme or acute episode. But in any case, it has been the case that it attributes roughly for 1/3 of the deaths worldwide, and especially in the EU, roughly 1 out of 2 deaths are attributed to cardiovascular disease. And when I say cardiovascular disease, it can be anything from heart failure to pathological hypertrophy and like a myocardial infarction. So this term kind of encompasses every one of these issues. Now, hypertrophy is a process that happens also in physiology in athletes or in pregnant women. So we have a physiological hypertrophy in these cases, which is a reversible process. Now, in the pathological version, which is not one of these cases, the progression of heart failure is an irreversible process. You have an enlargement and thickening of the heart, but eventually it becomes dilated, and it's incapable of supporting the body with proper with blood, practically. So the effects that it undergoes are changes both in physiology and metabolism for the heart. And especially, for instance, after a myocardial infarction, many patients will develop heart failure because there is usually an occlusion in some artery, and then that part of the heart that is no longer oxygenated. So those cardiomyocytes die, and the total number of cardiomyocytes diminishes. Now, that is the case because the cardiac tissue has very limited regenerative capacity. So this cardiomyocyte loss cannot be reversed. And there have been many theories as to whether there are any cardiac progenitor cells, for instance, in the heart, cells that can be used to replenish those lost cells. Now, as you can see in the heart, the cardiac cell types are many. It's not just the cardiomyocytes, as one would think. Actually, cardiomyocytes only account for roughly 1/3 of the total population of cells in the heart. It's a very heterogeneous tissue. You have endothelial cells, you have smooth muscle cells, you have epicardial cells, you have fibroblasts, and let's say potentially you have cardiac progenitor cells. But that's not just it. There are also subdivisions of these cells. So you have the endothelial cells, but you have arterial and venous endothelial cells. You also have capillaries. And then in the smooth muscle cells, you also have arterial and venous smooth muscle cells. You also have pericytes. So you can see that practically there are many subtypes of either endothelial or mesenchymal populations, and there are also specializations of cardiomyocytes, like the ones that give practically the signal for the pumping and the main work force of the heart, the actual cardiomyocytes that pump, that do the pump. So it's a very heterogeneous tissue. Now, if we want to identify, first of all, we want to identify whether we can find any cardiac progenitor cells. But in this process, we have to find out about all the different cell types. And to do that, we use an approach that uses single cell technology. Now, single cell technology allows us to uncover the transcriptomic landscape. And so far, there have been many approaches for the single cell technology, most of them focusing on the coding part of the coding aspect of the transcriptional landscape. So we have the mRNAs, and sometimes they also pick up some non-coding RNAs, like the long non-coding RNAs or the circular RNAs. Now, we've used one of the most sensitive techniques, which is the SMART-seq2, that was developed in the Karolinska Institute in the group of Richard Sandberg. And this allows us to have the full transcriptional landscape as far as coding, the coding aspect is concerned. But what about the non-coding aspect? And as Dimitris very nicely said in the beginning of this conference, of this session, that is where a lot of information rests. More than 90% of the information rests in this. So we have the small non-coding part, which we are missing. And to do that, there is another technique which has been developed again in Karolinska. It was the small-seq protocol, which allows the detection of microRNAs, snRNAs, small nuclear RNAs, piRNAs, but also tRNAs and rRNAs in a single cell manner. So combining these two, you can have the full transcriptomic landscape. And this is a process that is, we do this in the cardiac development. Now, why we do this in the early cardiac development? So if we want to find, let's say, cardiac progenitor cells, we have to do this if we want to see if there are any cardiac progenitor cells in the adult heart. We need something to compare them to. So we go very early in the heart, in the cardiac development, and we see, we try to identify the transcriptomic fingerprint of the cardiac progenitor cells during the natural development of the heart, because these are for sure cardiac progenitor cells. So to do that, we went to embryonic day 8.5 and 9.5. During these time points, actually, we have two anatomical structures, which is the first and second heart field. First heart field here you can see with red, and green is the second heart field. And these structures practically with looping and of course the expression of different genes give rise to all the cell types in the heart with contribution from another structure which is called the proepicardium. But these structures practically give rise to all the different cell types in the heart. And these two anatomical structures are characterized by the expression in these time points of different transcription factors, which is known from literature. And specifically for the first and second heart field, we have the NGX 2.5, which is a transcription factor, and we have the islet-1 for the second heart field. So by using two transgenic mice, one with the NGX 2.5 promoter coupled to GFP and another one with the islet-1 coupled to GFP, we can isolate cells that are expressing these two transcription factors in these time points. So we are certain that we're isolating cardiac progenitor populations. Now, how is the process of a single cell experiment? How does it look like? First is a tissue dissection, which is followed by dissociation, usually with an enzymatic process or mechanical dissociation. And then we have the addition of fluorescent antibodies in the single cell suspension with either cell surface or viability markers, followed by the actual FACS sorting. We use the FACS area 3 into a 384 well plate format. Now, how does it look like when we isolate them? These are practically panels from the FACS sorting. And on the left, we have the E8.5, and on the right, we have the E9.5. And you can see here we have the total cell population, and we do a singlet discrimination based on the forward scatter area and height. So we make sure that we just are picking single cells. And then by use of different viability markers like DRAG5 and DAPI, we make sure that we only select live and nucleated cells. We don't select any debris. And then you can see the GFP positive population that we pick up, either NGX 0.5 or islet-1. We know that already when we perform the experiment, depending on the condition. So we did that for 8.5 and 9.5, and we isolated cells in a 384 well format, and we submitted them for sequencing, for single cell RNA sequencing with the SMART-Seq2 protocol. And the workflow after that includes a lot of bioinformatic analysis. So we have the mapping, and then we're using one of the most established algorithm, which is the CEURAT version 3 for quality control and clustering. And then we are able to do cell type identification on those clusters, see what kind, because there is a lot of literature regarding which genes are expressed specifically in certain populations at these time points. So we use this information to make cell type identification, and then we can do reclustering on those specific clusters to identify subtypes. And this is practically a loop until we feel certain that we have identified all potential cell types. And eventually we do an anatomical verification using immunofluorescence. So this is how it looks like for E8.5, for instance. You can see here we have the second heart field cells and the first heart field cells, and also some cardiomyocytes. We have some endoderm and neural crest cells and nervous system we picked up, and pharyngeal arches. And how can we tell that these are practically these cell types? We go back to the genes. How are they expressed? NGX 2.5, indeed, most expression falls within the first heart field and spreads in the second heart field. Islet-1 is more restricted in the second heart field, and certainly not in the more mature cardiomyocytes, as you can see here. And then we have TBX5, which is very selective for cardiomyocytes, only expressed in this small part, with as well as cardiac troponin T, again a marker for cardiomyocytes and maturing cardiomyocytes. So we can use all these genes that we get from the single cell experiment to have a very concrete transcriptomic fingerprint at this stage. And then, as I said, there's anatomical verification. This is just to show you how it looks like. This is not an embryonic heart, this is a neonatal heart. But again, you can see, for instance, myosin light chain 7, which is an atrial cardiomyocyte marker, and myosin light chain 2, which is a ventricular cardiomyocyte marker, how they separate. And we can make sure that all our markers are very specific for the cells, the cell types that we are proposing. And the same happens when we go in higher magnifications for genes like endomucin, more restricted to the endocardium, FAB4 and PKAM1 marking endothelial cells. So now we have the microRNA aspect. Now the microRNA aspect has an inherent difficulty. And this difficulty practically is that there are no known markers that are specific for a cell type in cardiac development. Now, there have been many experiments done for bulk RNA and microRNA sequencing in the heart, but all those experiments give us an overview of what microRNAs are expressed in total. We don't know the specifics. We don't know where they are expressed. And how can we tackle this? Well, first of all, we can combine the SmartSeq2 single-cell transcriptional profile that we have and the SmallSeq single-cell transcriptomic profile. And now we have clusters in both cases. So we will use target prediction algorithms such as TargetScan Mouse and Diana tools. And this algorithm practically, by using these two algorithms with the SmartSeq2 single-cell transcriptomic profile, we will be able to have a prediction of what kind of microRNAs are expected to be present in a cell type, in a per cell type manner practically. And then we will overlay this information with a SmallSeq and be able to identify specific microRNAs per cluster. And so far, we have used the SmartSeq2 data, and we have also created a consensus database with all the interactions from TargetScan Mouse and Diana tools. We have 800,000 interactions from TargetScan Mouse and 200,000 interactions from the Diana tools. And we have created a consensus database, which we are now trying to see which are the expected microRNAs on a per cell type manner that will be overlaid with a SmallSeq data. Now, this whole research is possible only with the help of amazing colleagues, actually, in both the Agricultural University of Athens and in KI, in Karolinska, especially Mitsis, our PI in the genetics lab, but of course all the members of the group, Eleni, Katerina, Io, Athanasios, Thanasis, Louis and Eli, also Aspasia and Saradis, and all the collaborators from Karolinska Institute as well, which have helped make this project a reality. Thank you very much. I will take any questions.
So thank you very much, Stefanos. Very interesting. I mean, as I said before, this is a new kind of technique. It's very few that really have the experience of really using it, even though everyone would like to do. We have some space if people can ask questions on the chat.